This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 4, recorded on April 1st, 2011. Hi, everybody. I'm Vincent Racaniello, and this is TWiM, the podcast that explores unseen life on Earth. And we are recording on April Fool's Day, but we won't make any jokes. Everything that we say will be in seriousness. Joining me today to talk about microbes from Princeton, New Jersey, is Cliff Mintz.
Hello, Vincent. Good to be back.
How are you, Cliff?
I'm fine.
You're doing well? Hope so?
Yes.
The weather up here in the Northeast isn't so great. Cloudy. Rained a bit today. But I'll bet down in North Carolina, where our second host is, Michael Schmidt, it's a little better. Welcome, Michael.
Thank you, Vincent. Today I have the fortunate task of being at the South Carolina ASM branch meeting that's being held in Clemson, South Carolina. So I'm going to go off to a branch event tonight.
Oh, so you're not in Charleston, which is where we usually get you.
Nope.
How far is that from there?
It's about 3.5 hours.
Ah. Do you have Chamber of Commerce weather today?
Yeah, we do. It's actually beautiful and sunny and about 65 degrees, so it's a beautiful spring day. All the wisteria are blooming along the side of the roads, and the azaleas are in full bloom, so it's really quite spectacular. You never know how many shades of green there are until you come to the Carolinas in the spring.
Wow, sounds great. Well, here it's pretty gray, but we're not complaining. We have a new host on TWiM joining us today from the University of Wisconsin at Madison. She is a professor in the Department of Medical Microbiology and Immunology. And I'd like to welcome Margaret McFall-Ngai. Welcome to TWiM.
Thank you. Thank you very much for including me. I appreciate the opportunity.
Sure. And I heard it snowed today in Madison, right?
You know what? So we've had a lot of snow, and it was quite deep, after that huge snowstorm that came through, and it all melted away. And the Lake Mendota has begun to completely melt. And so we thought we were out of the woods. And all of a sudden this morning it started to snow again. So yeah, we're still in winter. I'm very jealous of North Carolina because it's completely gray and brown still here and white.
Yeah, well, that's Madison, as you know. You've been there a while, so the winters are tough.
And long.
So before we start talking today about our stories, I'd like Margaret to tell us a little bit about you and where you came from, your educational background and what you do. Where did you start out?
Yeah, actually, I started out, my training is in comparative animal biology, and so I'm not a trained microbiologist. So I started out at UCLA as a graduate student, but my PhD research was on how animals, quote unquote, remodeled their tissues to use luminous bacteria in their behavior. And so part of that has to do with rendering tissues transparent so that the light of the bacterial symbionts can shine through deep when they're in deep in tissues. So because I found the phenomenon of transparency fascinating, I went off and did 5.5 years of postdoc in protein biochemistry and biophysics. Learning about the biochemical basis and biophysical basis of transparency, working in, of all things, the eye lens at Jules Stein Eye Institute at UCLA. And then I did a second postdoc at Scripps Oceanography with George Somro, and I was working on enzyme adaptation. So I'm very interested in how animals are adapted at the biochemical level to do one thing or another. And so I started my first faculty position at the University of Southern California, and I wanted to bring the expertise of my postdocs back to the field of beneficial animal bacterial interactions. And I became aware that there was a squid in Hawaii for which you could raise both the host and the symbiont independently of one another. And it was a Vibrio fischeri, was the bacterium. And the animal was a small benthic squid that the cephalopod biologists had used to study cephalopod embryology in the laboratory. So I knew it was a great laboratory animal. So I started on that adventure, and I invited Ned Ruby to come in as a bacterial geneticist to develop bacterial genetics in Vibrio fischeri so that we could study aspects of the onset and maintenance of a beneficial bacterial symbiosis. So I was at USC, got tenure there, worked there, and then in 1996, we wanted to do some big genomics work. And so I approached the director of a place called the Kiwala Marine Lab of the University of Hawaii to find out if they had positions open. And it just so happened that a guy there, Steve Palumbi, was taking a chair at Harvard, and I moved into his position at the Kiwala Marine Lab. And I was there, and Ned also moved there to the University of Hawaii, and we were there for 8 years. And then University of Wisconsin-Madison had a cluster hire in the field of beneficial animal-bacterial interactions and invited us to apply for the positions here. And so we moved here, and so there's a group of us here at Madison that studies beneficial animal-bacterial interactions. And I've been really interested in, because my training is in comparative animal biology, about how, with our new knowledge about the beneficial roles of bacteria, how and why the trajectory of the history of biology is impeding us from recognizing how important this is, not just to humans, but all other animals. And so that's a little bit of a hobby of mine. I speak a lot to various places about beneficial animal-bacterial interactions in general and their evolution.
Sure. On my other podcast, we've talked about it in terms of viruses as well. And so this is something we should talk about on a future TWiM. We should have you talk about your work and to give it as a way of background. How did you like moving from Hawaii to Madison?
I know, everybody asks me that, of course. So I actually grew up in Southern California. And so I just looked at it as a tremendous adventure, an opportunity to live in seasons. I had never lived in seasons, and it's really a lot of fun. I have to say that I am still an affiliate faculty at the University of Hawaii, and my study site is still in Hawaii, and I have a squid facility here in Madison. Where I have squid. So if anybody ever comes by, I'm happy to show them the facility. But I do go to Hawaii 3 to 4 times a year, or somebody in the lab, not always me. Yeah. Usually I go once a year.
Oh, that's good. Not 3 to 4 times a year isn't bad. That's great.
Yeah.
Hopefully during winter.
Yeah, right.
Yeah, I'm the one that goes in the winter. Yeah.
I talking to Chris Kondayan of ASM earlier, he mentioned that you had done a Meet the Scientist last year or 2 years ago. Yeah. I think that's right. So if listeners want to hear about what you're doing before we talk about it here on TWiM, they can check that out. We'll put a link for it in the show notes.
Okay.
Well, today we are not talking about beneficial bacteria. Unfortunately, we're talking about foodborne illness, and I happen to notice in scanning my literature sources over the past week or so, two foodborne outbreaks across 8 different states. So I thought this would be a good entree to foodborne illness. And then Cliff told me that that's something that he had worked on at UW, right, Cliff?
Yeah, I actually got my PhD thesis in the pathogenesis of Salmonella gastroenteritis.
Wow. So this is perfect. In fact, one of these is a Salmonella outbreak.
Yes.
So I found some articles on CIDRAP, and if no one knows about CIDRAP, Center for Infectious Disease Research and Policy, and I use it to trace what's going on in infectious diseases. They have a wonderful collection of articles, and they also have daily updates on what's going on. So I found an article about this 2 foodborne outbreaks in 26 and 8 states. That was from March 24th. And then there is an update on the CDC website. Actually, the CDC site is quite good. And I thought let's talk about the Salmonella one first. And I have just questions here because this isn't my area, of course. This is called Investigation Update, Multi-State Outbreak of Salmonella Panama Infections Linked to Cantaloupe. So on this CDC site, they have a wonderful case count map. It's mostly western and northwestern states. There is, I think, Maryland and Colorado, aside from that. And these are Salmonella Panama. So, Cliff, what is Salmonella Panama?
Well, bacterial nomenclature has changed so much since I've been working with Salmonella, but essentially Salmonella Panama is a serotype of Salmonella Enterotoxogenic, and it's a serotype of the species Enterotoxogenic. And everything is now, as far as I can remember, a serotype of Enterotoxogenic. But back when I was doing my research, I was working on Salmonella Typhimurium, which is actually classified as Salmonella Enterotoxogenic serotype Typhimurium.
Okay.
So there are common serotypes that infect foods, and there are others that are not so common. And the Panama serotype is not a very common cause of food poisoning or gastroenteritis as Salmonella goes.
And that's probably worthy of a digression right now, Vincent, about this nomenclature about pathogens. Pathogens are often split, and they give all these unique names, which is the bane of existence of our poor medical students who are trying to remember the significance of these things. And as Cliff so eloquently said, this is basically Enterotoxogenic,
Mm-hmm.
and it's because these specific, if you will, substrains cause different maladies or are associated with different food products that you can actually trace them to. They often were given the status of a unique species because they were typically associated with a food or a geographic region from which they were isolated. And so, if we were talking about soil microbes, they would not be of that significance to the general human population, and so they would only have one name. And so you shouldn't be intimidated by all the genus species names, other than understanding that what Salmonella is noted for in almost all animal species is literally causing non-bloody diarrhea. Mm-hmm. And the associated cramping that goes along with that particular illness, and the type of cramping that humans can actually describe associated with it. In animals, they obviously don't describe the cramping.
So, are Salmonella infections all foodborne, or are there non-foodborne infections?
Well, there are the majority of cases. So, going back to the pathogenesis or the progression of the disease, there are two main Salmonella branches. Everybody's pretty much has heard of Salmonella typhi that causes typhoid fever, and most people remember this figure, Typhoid Mary, who actually did exist, who was a carrier of the typhoid organism, who, because obviously Salmonella lives in the gastrointestinal tract. It's not a normal inhabitant of the gastrointestinal tract of humans, so it gets in there and it causes disease in the gastrointestinal tract. And obviously pretty much the only way you could transmit it is through fecal contamination per se of a foodstuff, or transmitting it from one person to another in material that has been handled by somebody who's a carrier who has not washed their hands very well after going to the bathroom.
This isn't the same strain then, or phyllotype as what you find in birds and reptiles.
No, it's not. Salmonella typhoid, which is not what we're talking about. I want to distinguish it because Salmonella typhoid fever is really a septicemia that is a very serious disease. And if it's strictly associated with humans, and it's only transmitted amongst humans. Now, that's contrasted with the Salmonella gastroenteritis that we're talking about, which is caused primarily by Salmonella enterotoxogenic with all of the variety of serotypes. So in this case, Salmonella enterotoxogenic is a very commonly associated with animals. Most people talk about association with chickens and other farm animals. So what happens in Salmonella gastroenteritis is that the food material is actually contaminated by usually fecal contamination, and then the food is contaminated and then mishandled in such a way so that the salmonella organisms that have been introduced into the foods, they grow out to relatively large numbers. Then the infected food is eaten by somebody unknowingly, and because of the size of the inoculum in the infected food, and this is really important, it takes roughly 10 to the 6th to 10 to the 7th colony-forming units of salmonella to initiate a gastroenteritis. This is in contrast with another serious gastrointestinal disease known as shigellosis, where anywhere from 1 to 10 organisms is sufficient to be introduced into the gastrointestinal tract to cause disease. So the food, unlike Shigella, where it only takes 1 to 10 organisms, the food needs to be abused. There needs to be a situation where the food is not handled or stored appropriately, and the temperature is sufficient to allow the salmonella to grow. So in many cases, what will happen is that you'll hear about an outbreak associated with raw eggs or improperly cooked poultry. Particularly with turkeys, they say you have to cook it long enough to kill, the pathogen that's most likely to cause disease, which is salmonella. So if you don't cook the food or at a high enough temperature, so in most cases it's got to exceed 160 degrees Fahrenheit, the bacteria are not sufficiently killed, and the higher the heat, the more rapidly they're going to grow. So if you undercook a chicken and then let it sit, it's just there's teeming with bacteria because they haven't been killed. People then eat the infected foods, and they come down with salmonella 3 to 7 days after they ingest the food.
So in the animals that provide the salmonella, does the bacteria inhabit their intestinal tracts, their normal flora in chickens and so forth?
They are commonly associated with it. I don't want to call it, and Margaret, if you know differently, I don't want to call it part of their normal flora, but they're regularly transiently infected, and there can exist carriers in animals as well.
My understanding was that at least in some reptiles, that salmonella is part of their normal microbiota, and chickens are birds are reptiles. So I don't know about birds particularly, but yeah.
They're commonly associated, like the turtles, the little turtles you'd buy in the pet store. There was always this big scare of handling the turtles and stuff. I'm not so sure that they're normal inhabitants, but they are frequently associated associated with these animals. So really, at the bottom line of all this is that there's fecal contamination introduced into the foodstuff at some point to yield sufficient numbers of salmonella to be ingested, and then they actually establish an infection in the gastrointestinal tract where they actually invade the gastrointestinal mucosa pretty much through the epithelial cells into the lamina propria of the gastrointestinal tract, and they're pretty much contained without getting bloodborne most of the times. Yes.
What is happening between days, between the time that you ingest them, this 10 to the 6th to 10 to the 7th, and the 3 to 7 days when you actually come down with symptoms?
You're actually developing the gastroenteritis. The salmonella are becoming locally invasive, and then if you go back and part of the earlier show notes with Brett Finley's elegant animations of showing how salmonella interacts in the intestinal tract with the type 3 secretion apparatus. Yes. They go through this very elaborate opera of injecting proteins into our cells and going through this invasion process where you get membrane ruffling, and you're effectively causing an inflammation of the gastrointestinal tract, and that takes a particular set of time. And Cliff is right on target, and it's all about the initial dose that you receive as to how quickly you will go through this development of symptoms, because it is a dose-dependent activity in terms of how quickly your gastrointestinal tract is going to react to the microbial invasion.
The reason why the dose needs to be so high is that you're taking food in through your mouth. It's going through the stomach. So there's a very large kill because they're not very resistant to pH. So you have to take in a large number, and then the remaining viable members of the inoculum that get into the intestinal tract, they colonize the distal small intestine, primarily in the ileum. And it takes them a while to colonize. It's not really clear how they associate. It's probably some outer membrane protein, maybe some fimbriae or pili that facilitate the attachment. They have to grow out and then begin to invade through a variety of different mechanisms. And it's largely a self-contained infection within the mucosal layers of the small intestine. However, the reason why I went on to that long diatribe about typhi is that in immunocompromised and younger and perhaps older patients, the salmonella can actually enter the bloodstream in rare cases. And Salmonella gastroenteritis, in some cases, can lead to septicemia and death. So it's pretty much a localized infection, and it usually doesn't cause typhoid-like symptoms. And the interesting thing that people in the field talk about is that in contrast with Salmonella gastroenteritis, which is primarily a one-bucket disease, as we say in the food microbiology industry, as compared with staphylococcal food poisoning, which is a two-bucket disease. Yeah, coming out both the mouth and the back end, whereas Salmonella and Shigella are primarily coming out the back end with very little nausea. I forgot what I was saying because I got this whole vision of the two buckets, but so in typhoid, to the contrary, a symptom of typhoid fever is constipation,
as opposed to the non-typhoid, which is the famous diarrhea.
Right, and the interesting thing is, and just as an aside, I worked with this organism for close to 4 or 5 years, and one of the biggest things about the difference between people that work in Shigella laboratories, the organism that takes 1 to 10 to cause disease, versus Salmonella is that typically people in Salmonella labs do not infect themselves because you almost have to drink, a culture of Salmonella, to get yourself infected. Whereas Shigella, if you're very, very lackadaisical or not fastidious about your hands and where your food is, you can easily get infected with Shigella. So they kind of, if you look in the literature, they kind of downplay the severity of Salmonella infections, whereas Shigellosis, or it's more commonly known as dysentery, is a serious enteroinvasive organism that's ripping up the gastrointestinal tract, and you could die from blood loss and invasiveness. But I have to tell you that anybody, and there was a fellow I went to graduate school with who somehow inadvertently, he was working in Clostridium, but he was helping me do experiments, and somehow he got infected with Salmonella. And what you'll see is that it's not that big of an infection. It goes away. But he said if you're infected, truly infected with Salmonella, and you have Salmonella gastroenteritis, you're on your back for a week or more, and it's a pretty devastating infection. It's not like it's not like traveler's diarrhea where you recover very quickly.
You have to take fluids, basically. That's the treatment.
And antibiotic treatment is not effective because it's so localized to the intestine. And if you're giving antibiotics orally or intravenously, if the organism is not, in a focus, that's accessible by the blood, pretty much antibiotic therapy is contraindicated for Salmonella gastroenteritis.
An interesting aside is I did a bit of research on the medicinal use of Bacteriophage, and some of my colleagues from the Eliava Institute in Tbilisi, Georgia, actually have a preparation for non-typhoidal salmonellosis in which they give you a phage prep. You drink the phage prep, and it literally cures you of your Salmonella non-typhoid based diarrhea because the phage are actually delivered into the system. And in this month's, in the March issue of Microbe, there's an interesting article on phage therapy, or the medicinal uses of bacteriophage. So I thought that would be an an interesting aside, as Cliff was talking about treatment and Vincent was talking about antibiotics, there are other antimicrobial solutions to effectively go after this. And if you're flat on your back for a week, you'll almost do anything, literally taking phage preps that come out of the river outside of the Institute at Eliava.
But here we don't use phage therapy in the U.S., right?
No, we don't use phage therapy in the U.S. It has been tried experimentally back in the early 20s of the previous century. And Eli Lilly actually developed some ethical pharmaceuticals based on phage, but they quickly fell out of favor as widespread use of other antimicrobials came into vogue.
I suspect that there's going to be a big comeback.
Yeah, you think so?
Phage research over the next couple of years because we're running out of antibiotics. But that's another discussion we're going to have.
I get a lot of questions on TWiM about using phage for therapy. So,
and the issue with, not to diverge too much, is that the issue with phages is that with phages there's in the case of Salmonella, there are multiple numbers of phages. So unless you really know what the phage type of the strain is, you're going to have a multivalent cocktail of like 3,000 phages.
Right.
And that's effectively their solution.
Right.
And without getting too much into the phage therapy, it's extremely complicated to develop an ethical pharmaceutical based on phage simply because of the lysogeny issue. Many of the bacteria are already lysogenized with phage, and so they become immune to many of the preps. So combinatorially, it becomes very complicated to make an ethical pharmaceutical that will hit the spectrum of illness that you may be having, because you're essentially presenting empirically. And that's effectively how this patient cohort in the stories that we're going to discuss today presented. They presented with foodborne based illnesses. And I guess to put a fine point on it, each year, these are 1999 statistics, we have about 1.4 million people. That's the CDC estimate that actually will develop a salmonellosis, non-typhoidal diarrheal-based syndrome. And so it's pretty significant in terms of, you consider the number of Americans actually that will acquire a disease. And the CDC estimates that this is underreported by 38-fold. So in the case of the cases that we're discussing today, where we saw the cantaloupes infecting, or in the case of this bologna incident with Salmonella Panama, we had 12 patients from 4 states. They recommend you multiply those 12 patients by 38 to get actually the true case attack for this particular bologna outbreak.
Do you have any sense whether or not it's increasing in incidence over the last few years, or is it?
Interestingly enough, Margaret, Salmonella has been in the news more in the last 5 years than I've ever seen it before.
Yeah, so because, of course, I'm thinking about the changes in the microbiota that we've been talking about in various places and meetings that I've been at.
Well, the really interesting thing about it is that if you look at it, there were major outbreaks in peanut butter like 6 months ago, and before that there was a major outbreak with it was in the news like 2 years ago. I mean, it was all over the news, and it was the Salmonella outbreak. And as much as I'd like to say that it's an evolutionary, change, essentially what's going on is that the ability of people to understand how to handle food and the water supply that is being used to handle food, or the quality control, quality assurance testing of the facilities, slaughterhouses, or where things are put together, has really been lax, because rather than go to major suppliers these days, a lot of the stuff is being sourced from smaller providers, even people even operations in Mexico, or even out in the Midwest where there have never been FDA inspections of their facility. And unless you really understand microbiology and the fact that you have to be, when you're making food, the animals have to be far enough away from where you're preparing the food, and people that are preparing the food need to follow current good manufacturing practices surrounding the preparation of food to be shipped to consumers.
So this one is a cantaloupe-associated outbreak. And so they say, most of the people who became ill reported eating cantaloupe.
Right.
So, and they haven't yet proven that it's the cantaloupe, but
right.
The cantaloupe came from Guatemala.
Right. That's right. So Panama, Guatemala, it could be Panama City too, Florida, but
So how do you get salmonella from cantaloupe, Cliff?
Well, I would say, if I was a betting man, that this is what I think, but if it's not, we don't know for sure it's a cantaloupe, but I would bet that the cantaloupes were prepared before they were shipped with washed in contaminated water. And I suspect that they were shipped not in a non-refrigerated way from Guatemala, wherever it came from. And I think they were stored inappropriately. And then I think that if they were sold, whole, whatever, people that would cut through the rind and get to the flesh, that could introduce it. Or alternatively, if it was cut at the store and the knife introduced the bacteria into the pulp and they were not refrigerated or stored, at not the appropriate temperature, room temperature, and then put out in the store on an ice thing, that they grew out to sufficient numbers.
In this particular case, they were distributed at these wholesale clubs in 7 western states, and so there were 3 cantaloupes per mesh bag. And so, as Cliff said, if they were washed in unclean water that had the initial inoculum that may have been well below the threshold, then they went on the slow boat from Guatemala to the Western Distribution Center. They effectively grew, and many times in these wholesale clubs, they put out this green fruit at room temperature, allowing the microbe to effectively grow on the flesh. And then just imagine how you cut cantaloupe in your kitchen for breakfast. You cut it, and then you use the same knife to hollow out to get the flesh that you're going to actually eat. And oftentimes you don't clean the knife in between the initial cut and then subsequently touching the flesh. And so, again, it's all about how you dress out the fruit and in terms of using it. Many folks don't wash the outside of the cantaloupe before they cut it. And even if you could wash it, the question is will you have diluted the microbe that's attached to the flesh to a sufficient level such that when you cut it, you will not transfer those microbes to the flesh that you're going to immediately ingest. Because most people, when they prepare cantaloupe, it's just-in-time preparation. You cut the ripe melon, you slice it, and you present it for immediate consumption. Or you may place it in the refrigerator where the microbe will just idle in place until such time as you ingest it. And again, looking at the surface area of the rind that has the potential to get them, because most of these fruits are cosmetically clean, so they don't have mud or soil or debris on them, and so they go through a wash. But who's to say that they're the indigenous microflora has been reduced to a level? And again, the rind of a cantaloupe is very rough. It's almost a perforation, so it has much more surface area than you would anticipate just looking at it. When you compare a cantaloupe to, say, a honeydew melon, which has a smooth rind.
Impossible to clean a cantaloupe, really, with water, right?
So I have heard that there is some suggestion that salmonella and certain other microbes that through fecal contamination of the soil and so on can actually be taken up by the seeds and ovules and other portions of the plant, transported in the xylem or whatever and taken up. Is that not a possibility?
It's certainly a formal possibility, but I would almost go along the lines of, if you had a sufficient load of salmonella growing inside that very moist incubator of a cantaloupe, I think the flesh would be rotten, and you could actually detect a malodor associated with the fruit from the active growth of the salmonella. Spinach and other green leafy vegetables, where I think the majority of those studies have been done, I think are a different case because it's not as rich of a growth medium for the salmonella to multiply. If I was a betting man like Cliff, I would bet that the salmonella would amplify to an extremely high titer that you could actually detect it with your nose or immediately with your taste.
Right.
It's really interesting that, back so the where I trained, I did this research. The guy who I trained with was actually a bona fide food microbiologist, and there was always this sort of urban myth that if you leave not fruit salad, but tuna salad, potato salad out at a picnic, and then you should eat it because it would support the growth of bacteria. So I actually, there was an outbreak of something at some local supermarket. So I actually did the experiment where I inoculated ham salad, chicken salad, and potato salad that was bought from these stores and followed the titer of salmonella over time. And I was shocked at the results of the experiment. Now, this is a laboratory strain of salmonella that was isolated from a patient with Salmonella gastroenteritis. We must have passed it, several hundred times. But nonetheless, the salmonella just died in all of these preparations because of the pH of the mayonnaise.
Oh, my God. All these years I've thought, oh, my God.
Yeah, I know. So the thing about this chicken salad thing, it's like really funny. So I actually did the experiment, and ham salad absolutely does not support the growth of salmonella, probably because of the nitrites and stuff. But it was really funny. I just bring that up because salmonella is a pretty hardy organism. And you don't think, you don't think that, even a mayonnaise concoction would hurt it. And it really does. So, I mean, it just goes to the point that these things really had to be contaminated. There was no heating, of the cantaloupe. And as Michael pointed out, I mean, also another thing to consider is that if you use a knife or a cutting board and you don't clean the cutting board up and you just leave that sit at room temperature, and there was salmonella on the rind and it was all over the juices all over the place, you could also contaminate your entire kitchen. And that's why when people handle poultry, they always say, when you're done, wash the knife with soap and water and wash the cutting board that you cut the raw chicken on with soap and water to remove the possibility of salmonella contamination. Now, most people believe that all chickens are contaminated with salmonella because you're told to do that. And in fact, most of the chickens that are delivered to our tables are not, because they should be inspected by FDA, and they go through pretty rigorous inspection criteria. But that said, even if the bird, if you will, is contaminated with salmonella, if you cook it appropriately and cook it at 350 degrees Fahrenheit for an hour or an hour and a half, that will be sufficient to eliminate the potential load that may have existed on that chicken. So you're killing it after you cook it. So the key is that you have to wash the board of the knife because it's not going to be heated. So if there was any contamination, you definitely should do that. And if you're going to cook a chicken, or a poultry, make sure it's sufficiently cooked. Because if it's undercooked and there was a contamination, which you'll not see or smell or even realize that it's there, you can become ill that way.
So in the case of fruits, which you can't cook, you have to just extensively wash them. And for a cantaloupe, after you cut it up, you should wash it again. Is that the take-home message?
I don't know, because I've never heard of cantaloupes being a source of salmonella before. But anything. I mean, again, going back to this, it's frequently fecally contaminated water, or there are carriers, people like Typhoid Mary, but typhoid. But you can be a carrier carrier of Salmonella, the type that causes gastroenteritis, you can be a carrier.
Right.
So for those of you who don't know what a carrier is, it's somebody who's harboring an organism or pathogen that it's harbored by that individual, and the individual usually doesn't show obvious signs of being ill.
In this case, they haven't proven that it's the cantaloupe, so
right.
it may not be. But what do they have to do to address this? What will they do to see if in fact these infections were originated from the cantaloupes?
They'll probably do a spoof on that Saturday Night Live where they take the Basomatic and they'll literally take the cantaloupe and they'll homogenize the entire piece of melon. They'll homogenize the entire melon, and then either they'll use PCR to effectively ask the question, is it there? Or they'll do old-fashioned bacteriology, where they'll do plate counts on selective and differential media, asking the question, is Salmonella present? And if so, at what concentration? Going back to what Cliff said earlier, it's a concentration thing that will ultimately determine whether or not you will get sick from that melon. So ideally what you'll do is you'll take the melon, you'll slice it, you'll put it rind and all into a blender, you'll blend it up, and then you'll do serial dilutions and ask the question, what concentration do you get? And if it's past that threshold that is known to cause illness in a healthy adult, then you have implicated your melon in this particular process. Obviously, they have probably done this with epidemiological tracing, where they've asked everyone who has presented, so what do you all have in common? And I think what they all had in common is purchasing cantaloupes at the warehouse club that then, because the warehouse club sources its food from typically one common vendor, they were fortunately able to contact trace back where the melon came from.
Yeah, it's a single farm in Guatemala, in fact. So, I mean, I presume they can go back to the farm and say, hey, where did the salmonella come from? Was it from irrigation water or wash water?
Or from infected people, yes.
So they could fix that farm, maybe, but how do you deal with the fact that maybe next week they're going to buy it from somewhere else?
Well, typically what goes on, part of the FDA function, Food and Drug Administration function, is to actually, they're responsible for overseeing foods, drugs, and cosmetics. And essentially, it's up to FDA inspectors to go out to the chicken farms, to go out to the place where they're raising, fruits and vegetables that will be consumed, to do inspections. Obviously, the FDA is not going to routinely go down to Guatemala to inspect the farm there. They may not even know that the company had actually bought their fruit from Guatemala. But there are codes that the FDA, good manufacturing practices or regulations that FDA imposes on operations that manufacture and mass produce food. And as I recall, because of the outbreak, I'm trying to remember, I can't believe I can't remember, but the outbreak of Salmonella, I think it may have been in egg products, or it was actually maybe not a fruit. They actually went to, typically what you'll find is that they'll identify it through epidemiological methods where they think it is. And as Michael eloquently pointed out, they will identify that this is the place where it likely came from based on, bacteriology, based on PCR, based on genotyping, and showing that it is exactly the same from a molecular biological standpoint, the same strain. And they'll go back and then they'll start looking at the way things were done at the facility. And invariably, when there's an outbreak, if you go back to the source, it becomes very obvious very early on where the problems lie. And typically, there are all kinds of violations in terms of, handling the food, storing the animals, cleaning the carcasses, cleaning the equipment, that kind of stuff.
Right. So, Margaret, you had asked earlier about the salmonella actually going within the fruits, the flesh of the melon, right? I have this wonderful book here called Inside the Outbreaks, which is a history of the Epidemic Intelligence Service. And they have actually found salmonella within sprouts, alfalfa sprouts.
Yes, I remember that.
Within the actual flesh of. So there's nothing you could do. You can wash it all you want, and you never get it out of those.
That's right. Yeah, my understanding. We have a botanist here at UW-Madison. And Jean-Michel Launay is his name. And he actually has specialized in studying rhizobium, leguminous plant, nitrogen fixation, and so on, and mycorrhizae. But because we are in Madison here, he's also become interested in foodborne illness in plants and has spent a lot of time looking into this particular thing. And apparently, I guess from what he says, yes, right there inside of the flesh of the plants, you can get at least, like bean sprouts or whatever. And I think that the other, like I said, in this cantaloupe incident, in many, many different kinds of fruit, they found Bacillus species, Cytophaga, Acinetobacter, various things inside the ovules and inside the seeds.
Yeah.
But Salmonella, no, I'd never heard of that. And of course, people don't eat cantaloupe seeds. So that's very, very unlikely.
Yeah.
But it would be interesting to know in this instance if it was right there in the flesh. Now, I suspect that it's exactly what we're thinking of here. Is it coming from the outside and in contaminated machinery and whatnot? But I think we shouldn't discount the possibility that it might be right there in the fruit.
Michael, you were going to say.
Well, there's a workaround for this that's a bit controversial. It's been suggested in the past, and that's the irradiation of the food, because you can actually pass food underneath a strong gamma source, and since the news media has been talking about cesium-137 a lot and cobalt-60, you can, of course, pass food underneath a gamma source to kill the microorganisms, and it's fairly well worked out how much radiation that you have to impart. And the analogy is made that it's very similar to pasteurization. Pasteurization, recall, is a process that doesn't make the food sterile. It simply lowers the concentration of microbes to a level that is acceptable, and that's typically 30,000 colony-forming units at the sell-by date. 30,000 colony-forming units is typically the pasteurization level that a product like milk and any pasteurized liquid can have at the time of, sell-by X date. And so that's not sterile. So you could typically reduce the level of salmonella on the surface of the cantaloupe or even inside the cantaloupe by effectively passing it under an irradiation source in order to make the food safe. And in fact, the FDA actually, there are some fruits that we currently get from Vietnam, I believe, and Ray Ortega, one of ASM's wonderkins that helps produce this podcast, on one of his podcasts actually describes a particular fruit that is actually irradiated before it actually comes into the United States. So again, there's a workaround if this becomes a widespread problem. And the reason we don't presently irradiate all cantaloupe coming into the United States is because it's not a widespread problem, and we're able to eat the raw fruit with just normal hygienic practices. So I think it's good that the CDC is contract tracing this back to the farm in Guatemala. I think a little bit of epidemiological sleuthing will help them address the problem so that we don't have to go to the extreme of irradiating our food in order to make it safe to eat for the mass population.
Yeah, it seems like there's a low-tech solution, which would be a lot less costly in this case, right?
Yeah.
Okay, that's a good discussion, I think, on that. The other foodborne outbreak had to do with bologna, as Michael mentioned. And this is 14 people in, these are mainly eastern states, Maryland, New Jersey, North Carolina, Ohio, and Pennsylvania. And these were associated with E. Coli O157h7. It's a different bacterium. Cliff, do you know all about that one as well?
Yes, I do.
All right.
But I don't want to go. So the E. coli H157 story started evolving about 20 years ago, and I think many people will recall the outbreak, I guess, in a Jack in the Box, or I don't want to implicate a particular fast food vendor, but a lot of children ate these undercooked or tainted hamburgers, and then they got very, very sick, and some of them died. And one of the common symptoms was kidney failure amongst the people or the children that died. So E. coli, there are different flavors of E. coli. Typically, just for everybody to understand, E. coli is a normal inhabitant of the gastrointestinal tract of almost all mammals that I know of. We have tons and tons and tons of E. coli in the human gastrointestinal tract. However, there are variants of the helpful or the normal E. coli that acquire the ability to cause disease amongst people that ingest these organisms, this special type of E. coli. And within the E. coli realm of infection, there are two distinct or even three distinct types. There's something known as Enterotoxogenic E. coli, which are E. coli that produce toxins in the lumen of the intestine that cause a dysfunction of the ion transport, and it causes diarrhea. These kind of guys are very similar to the Vibrio cholera type of outbreaks that we read about from time to time.
So these E. coli don't go into the lining of the mucosa like Salmonella, right?
They have known attachment factors, primarily fimbriae or pili, or even outer membrane proteins that interact with receptors in the gastrointestinal mucosa. They're not invasive. They just kind of hang on, and then they produce toxin, and the toxin exerts its action through a number of different mechanisms, which really are not important to this discussion. However, there's also another type of diarrheagenic or enterococcal E. coli, not enterococcal, but cause gastrointestinal disease, and these are known as enteroinvasive E. coli. And in the business, we consider enteroinvasive E. coli pretty much Shigella that has some E. coli characteristics. So, if you will, enterotoxogenic E. coli are what we call Shigella that have assumed some E. coli characteristics. So the disease that they cause, the E. coli 157 that we're talking about, is perhaps the most well-known villainous strain of enteroinvasive E. coli that actually colonizes the intestine after you ingest the contaminated food. And like Shigella, it requires much lower numbers to induce the infection in the gastrointestinal tract. And these guys, like Shigella, will cause massive destruction of the distal gastrointestinal tract. And they give you a syndrome known as dysentery, which is interpreted for the lay people as bloody stools. So typically, the distinction between Salmonella and Shigella or enteroinvasive E. coli is that if you look at the diarrhea stools, you'll see that there's no blood in the Salmonella stools, but there's massive amounts of blood in the Shigella and enteroinvasive strains of E. coli.
Cliff, what does the O157H7 refer to?
That is a serotype of the enteroinvasive E. coli. Now, what makes some enteroinvasive E. coli more dangerous than others is on top of the enteroinvasive ability to actually the genes that encode proteins that allow it to invade deep into the intestinal mucosa, strains like the OH157 also produce toxins that are similar to some extent to like the toxins produced by toxic shock strains of Staphylococcus aureus. And these toxins are cytotoxins that actually interfere with the physiological function of many organ systems, most notably the kidney, the kidney or urogenital tract in humans. And that's why this particular subtype or serotype is so dangerous.
If I could interject here, one of the fun facts about 157H7 is that while it's still called E. coli, its genome is 40% different than the standard E. coli many of us work with in the laboratory. And if you think about it, that means that if humans and chimpanzees were that much different, chimpanzees would actually be classified as humans, because as we all know, chimps and humans aren't that much different.
That's right,
from a genomic perspective. And the shorthand I always remember about O is the cell wall antigen number, and the H refers to the flagella antigen number. And this, again, will give the general listener an idea that these microbes have many different antigenic signatures that our immune system reacts to. And by virtue of the fact, if you can imagine, there's at least 7 different types of flagella, you can understand how you can be reinfected over and over again with these things as food poisoning agents, and that you don't become, if you will, immune to them. And immunity is an interesting concept when you consider food poisoning, because how you prevent food poisoning is typically easy, good hygienic practices. But then if you're always talking about, your immune system being so clever, then why is it that we can get food poisoning over and over again? And the short answer is because the bacteria are constantly changing their spots to confuse our immune system.
So, Cliff, you mentioned that this O157 is a relatively recent introduction. So where did it come from?
That's a good question. I guess, and if you look at the genomic sequence data of Enteroinvasive E. coli and compare it to Shigella, they're very, very similar. These things have a tendency. It's a very good question. Where did it come from? I suspect that either the flagella type or the O antigen type or something gave this a selective advantage as a prevalent type. It could be that there was a selective advantage that the OH157.
But I mean, do they come from a cow, for example, because it's mostly beef?
E. coli are associated with all mammals, and E. coli predominantly, it's like cattle. Yes, cattle. There are strains that are specific. There are bovine-specific E. coli. There are porcine-specific E. coli. I mean, it's really hard to say that, it depends on the type.
Yeah. So this was in bologna, which, of course,
comes from
has beef in it, I guess. Well, I don't know what's in it.
Right.
Yeah.
Well, I mean, what's in it could be a combination of beef and pork.
Right.
Well, no, it's a semi-dry beef sausage.
Okay.
Okay.
So it's beef. But what's interesting about this is that typically in this fermented food products, you would expect the lactobacilli, and the way that these products are preserved is with a rapid drop in pH. And so maybe there was a problem with the starter culture that was used to start this Lebanon bologna.
I see.
And so consequently, you didn't drop the pH to a level that was sufficiently low to prevent, and again, it goes back to dose. And while this is not salmonella, you need far fewer E. coli 157H7 to cause disease. And so the pH may not have dropped, and then the E. coli is hiding amongst the other proteins in which you effectively had the protective higher pH that allowed it to survive. And the way these fermented meat products work is you want a low pH to effectively preserve the meat. After all, that's why sausages became so popular before refrigeration was in widespread use, because it was a way of preserving the protein content from a slaughtered animal for a long period of time.
Right. Yeah, these are from a producer in Pennsylvania, so this is not a foreign producer. And so 23,000 pounds of products probably contaminated. And it's probably a production issue, as you say, Michael.
Or it could have been, Vincent, it could be a production issue, or, who knows what their process is. They may have added filler meats, or They may have used contaminated agents while preparing. You know what I mean? It's hard to say exactly where the contamination.
Would this production of this product be regulated by the Department of Agriculture or something?
Or
right?
It'd be FDA.
It would be.
Yes, it would be FDA. FDA is responsible for all food that's manufactured.
So they have to go in here and investigate and see what's going on.
They're going to go in and they're going to take the place apart and look at their process. Clearly, these people, I mean, I don't know how long they've been in business, but if you're in business and you are manufacturing food for sale, FDA is obliged to inspect your facility and point out to you what is right and what is wrong with your process. And it could be that, I don't know how long they've been in business, but if they've been in business for a while, their process seemed to work. Nobody got sick before. Something changed in their process, whether that's where the meat is sourced, whether the starter cultures were contaminated, whether they're padding their final product with filler that's contaminated. So there's a whole host of things that only a epidemiological and FDA inspection would enable them to figure out what went wrong and how we're going to fix it. And until they fix it, they're not going to allow this manufacturer to sell product.
I would look at the microbiology first to ask the question about their starter culture, and typically in the manufacture of sausage to ask the question, how fast does the pH drop, and did the fermentation do what it needed to do in the time frame? And again, going back to the production method, it could be something as simple as the incubator that was used to do the initial fermentation to set the pH correctly was just off.
Or alternatively, they because they were under pressure, they shortcutted, and that could also be responsible. So the thing is that it's really hard to say, but I agree with Michael. I mean, with that many pounds, there had to be a pretty systemic issue, right? So either the water they were using to compound the stuff in the final process was highly contaminated, or they took shortcuts to meet commercial demand.
Well, we'll follow this and see what happens, and when they reach a resolution, we'll talk about it. Just in way of closing this discussion, Michael has a nice article in Emerging Infectious Diseases, which summarizes food-related illness and death in the U.S. As of 1999, and it has a lot of good information. So we'll put a link to that in the show notes. I'd like to read a couple of emails from our listeners before we wrap up this episode. The first one is from Eric, who writes, I just stumbled upon your podcast last week and wanted to say I enjoy all of them. While listening to episode 1 of TWiM, I began to think about the significance of finding human DNA in a bacterial genome. While this discovery is certainly very exciting, I do have one question. Doesn't the nature of lines make this discovery somewhat less remarkable? They are reverse transcriptases, which are built to copy and insert themselves into host DNA. I think this discovery may be due to the fact that line elements are very mobile sequences, and N. gonorrhoeae may be the victim in this case. What are your thoughts on this? And would you feel different if the DNA found in the bacterial genome contained a gene for a membrane protein or other random human gene instead of a line?
I have a short answer, or a two-word answer: restriction and modification. The wonder immune system of the bacterium is restriction and modification. The bacterial DNA is typically tagged. Many of us take it for granted when we use the restriction part of the bacterial system called restriction enzymes. EcoR1 is, of course, the enzyme in E. coli that restricts or slices and dices foreign DNA when it comes in. And to the best of my knowledge, human DNA isn't modified such that if it is introduced into a bacterium, it would not be restricted by the enzymes within the bacterial cell. And you just have to go to any popular molecular biology catalog and you learn that BAM stands for Bacillus megaterium, and ECO stands for E. coli, and HINDI stands for Haemophilus influenza, to appreciate the wonder of the restriction system. That said, line elements are promiscuous, and that's likely how they probably got in, avoiding the wonder that is the restriction and modification system. And once the DNA is modified, it, of course, will be considered self and be stably maintained.
Any other thoughts on this?
I actually don't find it so unusual that it would be Neisseria, which is, I think of as a human-specific pathogen. There are a lot of things like that might be occasional or opportunistic pathogens, but as I understand it, Neisseria is restricted to humans, and so that's where I would look for such elements, is in the half a dozen or so very specific human pathogens.
I think that because it's a line, part of its function is to jump around. And so that's probably why we found them in gonorrhea. But the real question is, does it have a function? And we don't know the answer. So that is something we'll find out in the coming years, and that'll answer the question. Would I feel differently if it were a membrane protein? Perhaps. Apparently lines contaminate many sequences. I read a post which said that the global ocean database is full of human lines. So, yeah, maybe it is problematic, but they did demonstrate by PCR that the line was in the N gonorrhea genome, so it's probably bona fide. The next one is from Allie, who writes, TWiV, TWIP, and now of all things TWiM, it just doesn't get any better. As an adult back-to-schooler and beginning again science learner, I can't believe my luck at beginning my ed adventure into science and beyond during the era of Axis of Unseeable, the TWs. Once I listened to a TW discussion, which I suppose means this week, the book, especially those dratted CHs on microbe-human interactions and all the replication stuff, arg, comes alive. Here I was last week wishing you TW guys had a microbiology podcast, and poof, my prayers are answered. I thought my ears would pop out of my head as I listened along to TWiM number 1 and actually understood stuff. This makes learning fun. You guys have a somehow magical combination of hardcore geekism, humor, loopbacks to clarify things for the LILs, less learned listeners, and way too cool. All the heavy hitters seem to be getting together to chit-chat. Thanks, a 10 to the minus 7th mole or something like that. Clearly more to learn. Let's vote you folks along with Sal Khan as egalitarian educators of the year. By the way, the piece of human DNA stuck in there on Neisseria gonorrhea thing was really eerie, as Sal Khan of the Khan Academy would probably say. And the copper thing, why can't microbes eventually get around that like they get around everything else eventually? Temperature, salinity, pH, penicillin. Years from now, I'll realize this was a dumb question, but for now it has me wondering. Why don't bacteria become resistant to copper, Michael?
I think it's thermodynamics. I think it's the fact that all living things must pump ions for a living, including humans as well as bacteria, and they go out in pairs. An electron and, in the case of the bacteria, a proton go out together, and they must come back in. And once it hits the quantum river that is metallic copper, the electron falls in, and it's just like us trying to leap out of a swimming pool. We just don't have enough energy to leap out of the pool, and consequently the electron can't rejoin its proton pair to effectively go back in to maintain the neutrality of charge. Remember, the chemoosmotic form of energy generation requires a balance of delta psi, which is the change in charge, as well as delta chemical, which is the change in this particular case of proton, because it's in the case of the bacteria, they generate their energy by adding the charge potential and the chemical potential, pH and electron, together in order to generate delta mu, which is energy.
Okay. All right. Thank you, Ali. The next one is from Kenneth, who writes, just about to download TWiV when I saw you have actually released the first episode of TWiM on Microbe World, and I just can't stop jubilating. I think it's great we make people so happy.
Yeah. Who knew that science was such a happy thing?
I strongly hope this podcast would be successful as TWiV and TWIP, and I appreciate your help very much. You're really helping some of us, and I just can't thank you enough. I pray for long life, happiness, and good health for you, your family, and all your friends. Well, thank you very much, Ken. The next one is from Stephen, who says, here's a couple of ideas for signing off the show. So the first one is, another TWiM is added to the culture. And another TWiM enters the culture. Those are pretty good. We'll think about those, Stephen.
Clever.
And the last one I will read today is from Attila. Dear Twimmers, first of all, congratulations to Professor Racaniello and Racaniello Media Corporation for this new initiative. Well, ASM helps us a lot. I am a regular TWiV and TWIP listener from Brazil, and just when I had listened to all the episodes, you released this new podcast. Now I can only expect This Week in Vertebrates or This Week in Plants. Until then, let me make my first TWiM question. Many times I have heard the explanation that the smell that comes after the rain is caused by antibiotics produced by actinomycetes and other bacteria in a silent war for space. But I have never seen a decent reference to support this. People always say it, but do not cite any source. Is it true, and do you have a good source for this?
I could answer that.
Sure.
So I learned this back as a first-year graduate student in microbial ecology. And I don't have a reference at my disposal, but I learned that the smell after the rainstorm is caused by a secondary metabolite produced by streptomycetes known as geosmin. G-E-O, I'm not going to be able to spell it. It's geosmin, M-I-N. So that's what I learned. So I think that, streptomyces are an actinomycete, and it's a secondary metabolite. And in many cases, secondary metabolites are emitted from organisms that are capable of producing them to provide better competitive edge over their neighbors in the soil. Antibiotics were primarily thought of as those types of agents or other things that bacteria produce that are known as bacteriocins, which are similar to antibiotics, but unlike antibiotics, they only affect same species of bacteria. So I think that it's largely true, and I think we should find the reference for it.
Wikipedia, geosmin, Geosmin, which literally translates to earth smell. So if you just do a wiki on geosmin, it's full of references to streptomyces and the enzyme, and all of the references can be there if you just use geosmin, Geosmin, and Wikipedia will give you the structure and everything else you could possibly want.
And I have to attribute that to the University of Wisconsin, Jerry Ensign, the guy who convinced me that microbiology was really cool. Was responsible for that bit of information, which I've never forgot.
All right. And Attila ends up, thank you for making another fantastic podcast. I hope you inspire other researchers to do the same, or else Racaniello Corporation will end up having 10 or more podcasts, and this is a monopoly. I won't do 10. I can't do that many. But it would be nice if others did the same, but it takes a bit of work. All right, that'll do it for TWiM number 4. And remember, you can find us on iTunes, at the Zune Marketplace, or at microbeworld.org/TWiM, where you can play the episodes, download them, and look at our show notes. You can also get an app to stream TWiM to your iPhone or Android device. It's at microbeworld.org/app. If you like TWiM, do tell others about it. And if you use iTunes, leave a comment in the podcast section. It keeps us on the front page. Send us your questions and comments to twim@twiv.tv. I'd like to thank everyone for participating. Margaret is at the University of Wisconsin-Madison. Thanks so much for joining us.
Oh, you're welcome. This has been great fun.
And you will be back, I know, on the schedule. We have you down for a couple of weeks from now. That'll be great.
Right.
Michael Schmidt, the Medical University of North Carolina. Thank you, Michael. South Carolina. You don't want to move to North Carolina?
Maybe next week.
All right, South Carolina. My apologies. Thank you for joining us again.
You're welcome, Vincent. It's been great.
We missed you last week.
I missed being there. I was out on the West Coast.
Yeah, well, there's a void on that Friday afternoon at 4 p.m. I understand that. Cliff Mintz is at biojoblog.com. Thank you, Cliff.
You're welcome, Vincent.
We were the beneficiaries of your expertise on foodborne diseases today.
Well, I got my PhD in it, so I should know something about it.
Good, and we will follow these stories and see where they end up. I am Vincent Racaniello, and I'm at virology.ws. I'd like to thank ASM for their support of TWiM. Communications Director Barbara Hyde, and Chris Kondayan and Ray Ortega for the technical stuff that makes this work. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.